Hello everyone. I'm Dr. Jiji George, Professor and Head, Department of Oral Pathology, Babu Banarsidas College of Dental Sciences, Lucknow. So today I'm here to speak about tissue processing. Now, tissue processing is any treatment of the tissues necessary to keep them in a lifelike manner as much as possible, to impregnate them with a solid medium, as to give them enough rigidity to facilitate the production of sections for microscopy. So when I say lifelike manner, I mean to say that when a biopsy or an autopsy is taken from the tissue, obviously it ceases to be alive. So it's not a vital tissue. So we want to see what all are the changes uh, that were there in the living tissue, which is why we actually do a biopsy or an autopsy. So we would not want any amount of protein degeneration or denaturation to occur. So we want it to be as much preserved as much as possible. That is what I mean by the word keeping them in a lifelike manner. So this is done to impregnate them with a solid medium. That is, you cannot just cut a human tissue, and you cannot just cut it with a knife into thick sections, thin sections, and see it under the microscope. We have to cut it in paper thin sections, which are microns in thickness, and to do so we have to remove the water component which is present inside those little tissue pores inside the body, inside the tissues. So our first aim is to remove the water and fill it with something like a wax, which will go and fill those tissue spaces and make the tissue rigid enough for you to enable to cut it with a microtome knife. So that is the whole uh, concept behind tissue processing, making the tissue ready for sectioning. So that is the whole concept of tissue processing. So now let's go to what are the steps in tissue processing. The first step in tissue processing is fixation. So when I say fixation, I mean to say that you have to put it in something like put the tissue in something like a formalin or formaldehyde to ensure that the proteins are not denatured. For example, if we make an egg, so if you want to make an egg, you first break the egg and just put it on a hot pan, and you suddenly find that the egg becomes white in color or yellow in color. That's because it solidifies. So that solidification happens because the proteins get denatured. So heat facilitates this denaturing. So we basically don't want the tissue to putrefy. So we want to maintain those proteins in the same situation as they are. So that is how we fix them. So that is called as fixation using formalin. Now, which is why you always get a very different smell when you go to a dissection hall where you have your cadavers which are put in tanks, formalin tanks. So the next stage is decalcification. This step is admissible only if we get a hard tissue. So talking about, we being dentists, talking about tissues, mostly we get hard tissues in the sense that even if you get a tooth is a mixed kind of a tissue. It has a outer harder covering of enamel and dentine, and inside the pulp is a soft tissue. So we have to, if we have to, even if you have to study the tooth, we have to initially decalcify, that is remove the calcium from the tooth structure, so that we can section the tooth very easily using a soft tissue microtome. So decalcification is very important. Then comes dehydration. That is again removing the water component from the tissue spaces in between. After dehydration is over, we do clearing. That is, the water is removed, and into those spaces we inject xylene. We let xylene enter those tissue spaces in between. So that is called as clearing. Now, xylene is a clearant because it is miscible not only with water, which was earlier present in the tissue spaces, but also with wax, which we intend to introduce into those spaces later on in the next step. So now that step is called as impregnation. So in impregnation, we remove the xylene and then impregnate those tissue spaces with wax. because water and wax are not miscible with each other. That is the reason why we need an intermediate agent called as clearing agent, that is the xylene in between, which is miscible both with water on one end and the other end with the wax. So what xylene is going to do is it will replace the water from the tissue spaces, and the spaces will now be filled with paraffin wax. So it will get replaced when wax is pushed in. So that is called as impregnation. So once wax goes and enters all the tissue spaces in between, the tissue becomes very rigid. Then you have to just make a block of wax uh, so as it becomes more easier to cut the sections by holding it onto the microtome. That is what is called as embedding. So let's take each of these sections in detail. Okay, after embedding, we do sectioning of the tissues on the microtome, and then we stain it with whichever stain we have to use in the specific situations, and mount it with a mounting media. Only then your tissue is ready to be seen under the microscope. So when you get a biopsy of tissue, a living tissue, uh, you first fix it and you take it through these tissue processing techniques. Ensure that the tissue is rigid enough so that you can cut sections and see it under a microscope. So this is basically the role of a pathologist. And uh, first, when you obtain the specimen, you have to label it. That is, if at all your lab gets more than a lot of numerous tissue specimens in a single day, or maybe on different days. So what happens is, if there are certain names are very common, like for example, the word puja, the name puja is very common in India. So maybe one patient whose tissue came in today, her name is Puja. Another patient whose tissue came in today, her name is also Puja. But then one is suffering from cancer and the other one has a inflammatory lesion. So we cannot intermix or interchange tissues because treatments are radically different. So we have to label the specimen. You can't possibly write the patient's name on the specimen. So you give them a number. Suppose the first number would be something like a 01, and if the patient has come in 2020, you write 01/20. The second patient would be 02/20. So basically the name does not matter because the tissue is given a number, and it is the data is documented in separate registers or folders, so that you get to know which patient exactly has what problem. So that's the first step. And after that, we come to fixation. So the first step of fixation is coagulation of the proteins. That is, alteration by treatment is further treatment is reduced. Tissue is readily permeable to subsequent application of reagents. So as I said, we use 10% neutral formalin. We put it in into this formalin for more than 24 hours of time. So depending upon the size of the tissue. The bigger the tissue, the larger the amount of time you have to place it in formalin. Another point you have to remember is that the volume of formalin that you insert it into should be 15 times more of the specimen. That means the tissue should be very well embedded inside the fluid. This fixation is done basically to prevent autolysis and putrefaction. So in that picture, you can see it's a glass jar which we have closed it with a lid. That is because the formalin is volatile. It can actually also just be lost because so we have to put a lid on it so that it does not just evaporate away. So that is how you put the tissue in. Usually in dental clinics, you get very small tissues for biopsy on a routine basis. So even a glass jar would be okay to put those tissues in formalin. Now, the next step that we're going to discuss is dehydration. Now, so now in dehydration, you remove the water content in the tissue. So that's the whole point of dehydration. And so that, as I told you, water and wax are not miscible. So in between, you need to use something like a xylene, which is miscible both with water on one end and xylene on wax on the other. So the first point that you would do is you would introduce the tissue in increasing grades of alcohol. So alcohol is something which dehydrates the tissue. So you insert or embed the tissue in increasing grades of alcohol. When I say increasing grades, it has to start from 60%, 70%, 80%, and then 90%, or maybe absolute alcohol. So it cannot start from below 50% of alcohol, because it will rehydrate the tissue, because water content would be more. So in a 60 to 70% alcohol, there'll be less of water and more of alcohol. So as a result of which, in increasing grades of alcohol, when you put the tissue, what happens is your tissue will gradually get dehydrated. You should not put the tissue from 60 to 100 directly, because it might shrink. So we don't want that to happen. We want water to come out of those tissue spaces in a graded manner. So we use grades of alcohol. So as I said, time depends upon the size and density of the tissue. So this is how we use bottles to fill alcohol. We can label them as 70% alcohol, 80% alcohol, and or either write it as alcohol, absolute alcohol 1, 2, and 3, depending upon what are the grades of alcohol which you use in the lab. So the next stage is what it does. Basically, next stage is called as clearing. Now, clearing is where you use xylene. So as I said, xylene is miscible both in alcohol as well as in paraffin wax. So the best part would be when you use xylene, it basically clears out the alcohol and it lets the paraffin enter in the next stage. So that is called as a clearing agent. Now, a clearing agent will increase the refractive index of the tissue and makes it appear translucent or clear, which is why it is called as a clearing agent. Now, volume of the tissue, again, as I said, it should be 50 to 100 times of volume should be there of the clearing agent. In a small tissue, you can put it in a beaker. Larger the tissue, you have to increase the size of the beaker in which you put the tissue into. So 2 changes of xylene are usually used, and you keep the tissue in xylene for 2 hours each. So that is how you put the xylene bottle labels, again xylene 1 and xylene 2, so that initially you put the tissue into xylene 1 and then you put it into xylene 2. Now, what it does in infiltration is it helps penetration of wax, paraffin wax, into the tissue spaces. So that means the xylene has to come out from these spaces and wax has to enter inside. So it replaces the xylene or whichever clearing agent is used. So this provides rigidity to the tissue. As I said, we have to ensure that you are able to cut the tissue easily on the microtome as thin sections, paper thin sections. So 2 to 3 changes of wax for 2 to 3 hours at 55 degrees Celsius is needed. So you have to keep the wax in a molten condition and then you insert the tissue cassette into the wax. So how is that done? We either use tissue cassettes or we use Leukert's L pieces. So I'll show you pictures of Leukert's L pieces. And the block has to be cooled later. Once you make the block, then you have to cool it by using, refrigeration or cold water. Now, that is what you mean by embedding. Once after impregnation is over, impregnation is the whole idea of letting wax impregnate inside the tissue spaces. Once after you ensure that all the tissue spaces are impregnated by wax, you have to do is embedding. Now, what do you do in embedding? You actually take that thick piece of tissue in which already wax has been impregnated. You keep that insert it into a block of molten wax. Once after this molten wax will solidify, you can, to ensure more solidification, you can either freeze it or put it into cold water. So after that, the wax block is trimmed. Now I'll show you pictures of Leukert's L pieces. These two L-shaped structure of metal, these can be used in approximation to each other. And in between this, you can actually pour the molten wax. And into this molten wax, you can put the tissue. So what place the tissue in? So the tissue is actually embedded inside the molten wax. And the wax surrounding it will get solidified and you will get a wax block. That is how you make wax blocks using Leukert's L pieces. Now, this is just to show you how you can arrange the Leukert's L pieces in different sizes and shapes. This can be a larger shape. The same L pieces can be used in as smaller sizes as well, just to ensure that you can, this is how you pour the molten wax into, you heat the wax on a gas, and after that you can pour this wax onto the mold, and this will actually some amount of molten wax will flow out, but then by and large it will get um, it would get solidified, and you can embed your tissue into this block. Now you can see that we have also kept a label out here. This label will ensure to make you identify which tissue block it is. So these are other cassettes which are used these days. So these cassettes are basically used for embedding tissues, like we made, or Leukoid cell pieces are a older stuff which we used to use. Now we make use of these tissue embedding cassettes. Now these cassettes are made up of plastic or polymer, and you can see that there are so many pores, small pores in those cassettes. You can just keep the tissue with the label inside the cassette, and then insert the cassette in entirety into the molten wax. So what happens is all the wax will get inside and embed around the tissue, and you would have a tissue block made up. So that's how your wax block will look like. So you can either do it manually, the tissue processing can be done manually, or you can do it in an automatic tissue processor. So the automatic tissue processor basically are time controlled machines. You can put it for a 24-hour cycle or a 12-hour cycle, depending upon what your speed of tissue processing has to be. So each of these glass beakers that you can see, one will contain your dehydrating agents in increasing grades of alcohol. Then a couple of them would contain xylene clearing agents in one or two changes. Then the next ones would contain molten wax in one or two changes for impregnation, and then ultimately it will come out. So once after the tissue goes through all of these automatically, this is run by a motor, and it would actually rotate and go through each of these each of these beakers. Your tissue at the end of the cycle, preset cycle, is ready for um, section cutting. So that's how we get it. So this is again a very old machine, that is a histokinete. Here you can see it very clearly. The beakers, these are empty here. These two beakers are for heating up the wax. That is, you will have molten wax out here. Rest everything, grade by grade, you would have dehydrating agent and paraffin wax, or clearing agents and things like that. So that is about tissue processing techniques. I hope it was informative. Thank you for your patient hearing.